Hello, everyone. We have lots of stuff to do today, so let's get started. Lecture 8 is about heredity. So there's two parts to this lecture. We'll first start by talking about the process of meiosis, which is how we produce gametes. And then we will spend the second half talking about Mendelian genetics. Okay, so we'll be working on those genetics problem. So before we begin with this worksheet, maybe let's just quickly review some of the key things that occurs in meiosis. Right? So we're going to start with a cell right here. I encourage you to draw this with me. We have a germ cell. Germ cell. And germ cell is basically something that will become a sperm or egg. Okay? So undergoes meiosis to become sperm or egg. Okay. And it is going to be found in the gonads, found in reproductive organs or gonads. Okay. Testis in female, in male, and ovaries in female. Okay, so male and female gonads, and in there we are going to have these germ cells which will undergo meiosis to form the gametes. So for simplicity, let's just draw some chromosomes here. We will have chromosome 1 from mom, chromosome 2 from mom, and we will have chromosome 1 from dad and chromosome 2 from dad. Okay, of course, this is gonna be more complicated in actual human cells because we have 46 chromosomes in total. But to not confuse ourselves, I'm just going to show 4 chromosomes here. Okay, so this is going to be a diploid cell. Diploid. Okay, 2 sets of chromosomes. It means 2n. Okay, so over here we have 1, 2, 3, 4, 4 chromosomes in total. 4 chromosomes. All right, so I'll give you a moment to catch up. So just like mitosis, we will have to begin with DNA replication. So after DNA replication, we will have the chromosomes like this. Okay, so we'll have 2 chromosome 1 from mom stuck together, 2 chromosome 2 from mom stuck together, and same thing for the paternal chromosomes. Okay, now I should mention here, forgot to talk about this earlier. This chromosome and this chromosome, what do we call them? How should we, what's the proper way of referring to them? Okay, this one and this one. Can someone help me out, please? The chromosome 1 here from mom, chromosome 1 here from dad. What's the proper term for referring to them? Not chromatids. Chromatids would be when they're stuck together. Good try though. Anyone else know? These 2 and then these 2. Okay, so we have a couple of tries. That's okay. They are homologous chromosomes. Homologous chromosomes. Okay, so what does it mean? It means they're same size. Okay, same length. And same order of genetic information. Okay, so for example, just making this up, if the information is like eye color, blood type, earlobe type, then on the other one it would be the same order. Okay, eye color, blood type, earlobe type. And so it's going to be found in the same order, but not necessarily the same version. Okay. So on one strand, it might be like brown eyes, the other would be like blue eyes or something. Not necessarily the same, but the order of genetic information would be the same. Okay. And similarly, this one and this one, they are homologous as well. But this one and this one would not be homologous. You're only homologous to the same chromosome number. Okay, so you have one inherited from each parent. All right. So after DNA replication, you will have the identical copies stuck together at the centromere. That's what that black dot is. But let's just label it just for completion's sake. Centromere. Okay. And these ones, this one and this one, they are identical copies of each other. We call them sister chromatids. Okay. Identical copies of each other. Okay. Same thing, this one and this one, sister chromatids. But this one and this one, they are non-sister chromatids, right? They are homologous. Okay, homologous. Okay, non-sister chromatids. So in the meiosis, the homologous chromosomes, after they duplicate, they will find each other, okay, and they will form a set of 4 individual chromatids. Okay? So together, collectively, does anybody know what this is called? Like the whole set here? What do we call that? Yes, or as a tetrad. That is absolutely correct. Okay, tetrad. So how many chromosomes do we have here? Well, we started with 4. After you duplicate, actually it's still 4. Because of the way we count it, right? As long as they're stuck together, we call it as 1. Okay, so 1, 2, 3, and 4. Right? All right. What else happens here? Okay, so during this formation of the tetrad, right, you will also have what's called like crossing over. Okay, so I'm gonna put it right here. Choose a thicker pen. Tetrad. Crossing over occurs. What is that? Okay, that is an exchange of genetic material between non-sister chromatids. There is no point exchanging genetic material between sister chromatids because they're identical copies of each other. Okay, so you will be, the red one here will be exchanging information with the blue one. Okay, and so afterwards, what you're gonna end up having is this. Okay, just try to find some space here. I hope you're drawing this with me. Okay, so you're gonna have a little bit of blue here on the red one. Okay, now most textbook just shows like the end being exchanged, but in reality, you're gonna be having exchange material along the entire length. Okay, and it's not just restricted to this one. You will also have exchange of material on the other one, right? So like you might have some blue here, here. It's kind of everywhere, okay? But it's confusing if you do it like that. So most textbooks just show you a little bit of it, all right? And then you're gonna have the, you know, the blue one, and now have a little bit of red on it as well. Okay, so same thing for the small one, for number 2. You can have some blue one here. Okay, like that. Okay, so this part here, that's called the chiasmata, right? The cross, that's where the crossing over occurs. So what is the purpose of crossing over? Exchange of genetic material between ancestor chromatids. This generates genetic variation. Okay, that's the reason why we look different from our parents, right? Okay, we're not going to be like identical copies of them because of this crossing over situation. All right, then what happens is we're going to have meiosis 1. We're going to have the division. Okay, we're going to divide. And after you divide, you will have 2 cells. They probably are still stuck together. But again, for simplicity's sake, I'm just going to draw them separately. Okay, so you will have the sister chromatids will separate from each other. So you will have there. Okay, and then for the other one, again, this is a random event. You don't really know what's gonna go to where. Okay, but you will have this one there. Okay, maybe this one. And over here, you're gonna have some of the blue as well. Okay, so this would be meiosis 1. Okay, so now we have 2 chromosomes. Okay, and then there's no DNA replication in part 2, so it will just divide again. I always have this problem. My drawing just ends up getting bigger and bigger. All right, and then the sister chromatids will separate. Right? You might get this one along with the other one that has a bit of blue in it. Okay, and that means the other one will get the longer red with a bit of blue, and then the shorter one. Okay, over here, same thing. We don't really know. It's all random event. Okay, so you might have a little bit of red on this one, a little bit of red though. Okay, so you end up having 4 cells, and let's say these are sperms. It's got a tail to it. Okay, so this is meiosis 2 and 2 chromosomes. Okay, and it's going to be haploid. Right? So that's the whole situation here. Does anybody have any question? Make these things a little bit smaller, maybe. Go, sort of. Okay, there is a question. Give me a second. Just want to find the Zoom button. There we go. Are sister chromatids also considered homologous? No. Homologous by definition is the one from maternal and the one from paternal. Okay, sister chromatids are like genetically identical, so not homologous. All right, so there you go. If we keep track of the human cell, we start with 46. Okay, and then after DNA replication, right, still 46. Meiosis 1, 23, 23. And then meiosis 2. (23, 23). Okay. Yeah, that's that. Can I move on? You want to screen grab this thing? I don't know. It's not really good screenshot. They're a little bit small, but you know, if you want, you can take one. All right. Should put this as meiosis number 2. Okay, let's take a look at this then. We have to identify the meiotic stage. Okay, first one here. So you can see there are like actually 4 cells now, right? 1, 2, 3, and 4 cells. So that means we have gone through the entire process already. This is going to be telophase number 2. Anybody want to tell me what this might be? What phase might this be? Anaphase 2 is correct. Right? So you can see one cell here, 2 cells, and each of them is having the chromosome being pulled to the opposite end. That is anaphase number 2. Okay. This one is just one single cell, and the chromosomes are being pulled to the opposite end. That would be anaphase number 1. Over here, you can see the chromosomes are kind of out of the nucleus already. And that would be just one cell. So that's prophase. Prophase number 1. This one is a little bit tricky. Anyone want to guess? That is prophase 2. Yes, very good, Laura. Prophase number 2. You already have 2 cells, right? And then the chromosomes are kind of out of the nucleus, it's ready to be lining up in the middle. And then here you also see 2 cells, and they're lined up in the middle. So that is metaphase 2. Now, how do I know it's 2 and not 1? Well, because there are 2 cells, right? If it's just 1 cell, that means we have not gone through the first division yet. So it's still meiosis 1. And this last one here, you can see the 2 separate nuclei. Right? Not very clear that it is separating yet, but it probably will soon be happening. So that is going to be telophase number 1. Okay, it's just before the cytokinesis. All right. Are we okay? Any question? No? Okay. Next part. Humans, 46 chromosomes. This is diploid. Diploid means 2 sets of chromosomes, so 2n, diploid. And 2n equals diploid, 2 sets. And, n equals the haploid, 1 set. Number of pair of homologous chromosomes. So 23 pairs, right? After meiosis 1, chromosome number will be 23. We drew it out, right? Where was it? Right here. See, after the first division, right? If you started with 4, now we have half of it, right? Because there's the chromatids are still stuck together. Okay, and this is going to be haploid. So just N. Maybe I do it in a different color so it's not going to be confusing. So this is 2N. This is N. You know what? They're all going to be the same thing. So I'm just going to circle it. So diploid over here, haploid, and then haploid again. So 23. Okay. No homologous pair anymore. Earthworm. So if you have 18 after meiosis 1, that means at the beginning you have 36. Okay? It's doubled, right? Or 18 pairs. And then it's still 18 after the second division. No homologous chromosome. Okay. It's pretty straightforward now, I hope. 90, 45, 45, 45, 0, 18, 9, 9, 9, 0. Does anyone want me to explain any of these? Which one you want me to explain, Adriana? All of them, I guess. Okay, let's just talk about the earthworm and hedgehog. Okay. So you start with 36, right? So 36 chromosome, like chromosome 1 from earthworm father, and then chromosome 1 from earthworm mother, chromosome 2, chromosome 3, so on and so forth, all the way to chromosome 36, right? So that's like 1, 1, 1, 2, 2, 3, 3, all the way to 36, 36, right? So this is pair number 1, pair number 2, pair number 3, and then all the way to, oh, sorry, not 36, 36, pair all the way to pair 18, 18. Okay, so a total of 18 pairs. Okay, but 36 chromosomes in total, right? So if you count like 1, 2, 3, 4, 5, 6, so on and so forth, right? And then you will have like 35, 36. Okay, or you can just take 18 times 2. So in total, there are 36 chromosomes, 18 pairs. It's like saying this. If I find 36 individual shoes in the closet, and I ask you how many pairs are there, right? Then it's like divided by 2, 18 pairs. So same thing with chromosome, right? The one from mom, the one from dad. It's like the left shoe and the right shoe. Okay, so this is pair number 1, pair number 2, so on and so forth. Okay. So now what happens is after meiosis 1, well, first of all, you're going to double these things, right? You're going to duplicate them. So you're going to have X, 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 so on and so forth, right? Okay, they get smaller and smaller. Okay. But this is still going to be counted as 1 chromosome, 2 chromosome. So this doesn't change after DNA replication. Okay. But then after meiosis 1, these things are going to separate, right? Okay. So one of them will go to one cell, the other will go to another. One of them will go to one cell, the other will go to the other, so on and so forth. So essentially, you're taking the starting number and you're dividing it by 2. Okay, and then afterwards, the sister chromatids will separate again, and you will still have 18. Okay. Basically, if you start with a number, okay, we call that 2n. After the first division, you would end up with n. And then after the second division, you will still have n. This is meiosis 1. This is meiosis 2. Okay. So other than the first number, everything else is going to be half. Any follow-up questions? Okay, great. Complete the following chart related to the difference between mitosis and meiosis. Okay, number of divisions in mitosis, 1. Number of divisions in meiosis, 2. Number of daughter cells in mitosis, 2. And for meiosis, it's 4 sperms in males and 1 egg, okay, or ovum, right? That's what it's called, in female. Okay, plus up to 3 polar bodies. All right. So if you don't know what a polar body is, there is the starting germ cell in female. We call that the oocyte. Okay. The germ cell in female. It's going to undergo meiosis number 1, but you will have uneven cytoplasmic division. So one cell gets very little cytoplasm, the other gets most of it. Okay, because you want an egg to be big. You don't want an egg to be like tiny. You want it to be big, lots of nutrients for the baby to grow. And then this is going to split again, uneven cytoplasmic division. Okay, so you will end up having these smaller things called polar bodies. Sometimes these polar bodies will also divide and you get like 2 polar bodies. So 1 polar body, 2 polar bodies, or 3 polar bodies. But then one mature ovum. Okay, that's going to be released into the fallopian tube. Right? Okay. Daughter cells for the mitosis, it's going to be diploid, 2 sets. Haploid for the meiosis. Number of chromosomes in daughter cells, 46. For meiosis, half of it, so 23. Okay, prophase here and prophase here. For the mitosis, the homologous chromosomes, they do not find each other. Okay, so they'll just be like, after they duplicate, they'll just kind of like hang out on their own. They're not going to find each other. All right. Whereas for the meiosis, they form the tetrad, right? Remember? So there's going to be one here. Maybe another one. And they're going to do a little bit of exchange of genetic material, remember? Right there. Oops. Okay. So for mitosis, homologous chromosomes do not pair up. Whereas for meiosis 1, homologous chromosomes form tetrad. Okay. Crossing over occurs to create genetic variation. All right. Metaphase and metaphase 1. Okay, so in mitosis, the individual chromosomes lined up in the middle. Whereas for mitosis, for meiosis number 1, for metaphase number 1 rather, they will be lining up in the center. The tetrad will be lining up in the center. Okay. Now you can see over here, the paternal one is on top, and over here, the paternal one is at the bottom. It's all random event. Okay. This is called the independent assortment, right? We'll talk more about that later. So over here, tetrad lines up in the center, at the center, yeah, of the cell. Okay. So genetic relationship, identical, no difference. And this one will be variant. It's going to be different. Type of cells involved, somatic cells. Okay. Somatic means bodily. So bodily cells. Most cells in the body is capable of doing mitosis, except for like brain cells, heart cells, some of the oddballs there, but most cells can divide using mitosis. And this is germ cells, okay? Spermatocyte, a male, and oocyte in female. Function, can someone tell me why we do mitosis? Why bother dividing? Well, yep, growth is correct. Growth and development. Yep, and then repair as well. If you have a cut, you're gonna have some new cells, and you are going to, you know, fill that gap. And for what is this thing? This is meiosis. Why do we do meiosis? For sexual reproduction. Yeah, for baby, exactly. Okay. Sexual reproduction. That is it for mitosis and meiosis. Any question before we move on to the problem solving? We're gonna skip the break today, okay? My other class, we totally ran out of time because there's so many things to do. So I'm just gonna I skipped a break with them as well, and we still run out of time. So you know, we're gonna skip the break. If you need to go for a break, feel free. Just you know, watch the recordings afterwards. Okay. So any question? All right, moving on. State the law of independent assortment. Independent assortment, what does it say? The alleles of two or more genes get sorted into gametes independently. Of one another. Okay. Well, that's what it says. But like, what does it mean? An allele. What is an allele? An allele is a different version of a gene. Okay. So like you might have a gene for eye color, and the different alleles would be like blue eyes, brown eyes, hazel eye, green eyes. Okay. All the variations of that gene is called an allele. Okay. So which allele gets put into a gamete would be an independent event. Okay. Like I'll explain with an actual example. Okay, so first of all, let's talk about what does it mean by independent event. Let's say I have a coin, okay, and then I'm gonna flip these coins. I have 2 coins, 3 coins, okay. What are the possible outcome when I flip a coin? Head or tail, right? Head or tail. Now, if I flip the first one, okay, I will have 50% chance of getting a head and 50% chance of getting a tail. So if I flip the first one, let's say I get a head. What's the chance of getting a head for the next one? Anybody? I already have a head. What's the chance of getting a head for the next one? It's not a trick question. Anybody know? Do you understand my question? My question is that, no. I was saying, okay, this is the question, right? Okay. When you flip a coin, it could be head or tail, right? Okay. So what's the chance of getting a head in a coin? 50%, right? Okay, yeah, 1/2. Okay, you get 50% chance of getting a head, 50% chance of getting a tail. I'm saying if I flip the first coin and I get a head, what's the chance of getting a head when I flip the second one? What's the chance? Anybody? Yeah, 50%, right? It's still 50%. 50% chance of getting a head, 50% chance of getting a tail, right? Okay. So let's say I flip the first coin, I get a head. I flip the next coin, I get a head. What's the chance of getting a head again on the next flip? Yeah, also 50%, right? Also 50%. In other words, what happened with the previous coins have absolutely no effect on the next flip. Okay? Each one has its own independent probability. They are not related to each other. Okay? So let me give you an example of something that's related to each other, right, so that you can see the difference. Okay? Let's say I have a bag of marbles. It feels like we're having a math class now. Okay, let's say I have a bag of marbles, okay? And we have some red ones here. Oh my God, can't even color. There is a red one here. 3 of them is red, okay? And then we will have some purple ones here or something. Okay, 1, 2, 3. Let's just do 3 red and 3 purple, okay? And the bag is opaque, okay? It's like you cannot see the inside, okay? You gotta randomly pick out a ball. What's the chance that you're gonna get a red ball? What's the chance that you're gonna get a red ball if you randomly pick one out? 50%, right? Because 3 out of 6 is red, right? So you have 50% chance of getting a red. Okay. Now let's say I do not return that ball back into the bag, and then now I try to take another ball. What's the probability of getting a red now? Well, it's gonna be 2 out of 5, right? You understand? Right? So what happened in the first draw will affect the probability of the second draw. So that's an example of a dependent event. Okay, the probability of what happens next depends on what happens on the first one, right? Flipping a coin is an independent event, right? Meaning what happens in the first flip has no effect on the next flip and on the next flip. Are we all clear about dependent and independent events? Any questions? You know, having kids is the same thing, okay? I have 2 daughters already, okay? So if I have another kid, what's the probability of having another daughter? 50%. Okay? Again, my grandmother, right? I told you about my grandmother. It's hard to explain to her. She's like, you have 2 daughters already. Try to have another child. It will be a son. I tell her, no, it might not be. Okay, it's still 50%. It's gonna be the same thing. Okay, these are independent events. They do not affect the outcomes. Okay, as I have many interesting conversations with my granny. In any case, what does that have to do with what we are talking about? Well, we are talking about the independent assortment, right? So let's say, yeah, you can do IVF. Yeah, I think IVF you have some control. Apparently, off topic. Apparently, like if you take the sperm and you spin it down with a centrifuge, because the X chromosome is a little bit heavier than the Y chromosome, then theoretically the sperm that goes to the bottom is going to be bearing the X chromosome. So if you take that to fertilize an egg, you will get a daughter. Whereas if you take the sperm from the top, you will probably get a son. At least that's what I read, anyways. But yeah. Anyways, back to here. I was saying, let's say we have some chromosome here. Okay. Chromosome here. This is chromosome from mom, and this is chromosome from dad. All right, and I'm just gonna use the stereotypical color. That's the one from mom, and that's the one from dad. Okay, so there are like hundreds and hundreds of genes, right, on the chromosomes. But for what we're discussing, we'll just focus on 3. So let's say on this one, it's going to be big A, allele, small b here, and small c perhaps. And then on the father chromosome, it could be the same, it could be different. But to illustrate my point, I'm going to say they're different, okay? They could be the same, or it could be homozygous, right? Okay. So what the law of independent assortment is saying is that, okay, so let's say that we're going to produce, let's say this is male, okay, and we're going to produce some sperm. So what gene will the sperm be carrying? Well, is it going to get the big A or get the small A? We don't know. It could be big A, it could be small A. Let's just say it's 50% chance, right? So let's just say it inherited the big A, okay? Now, just because the big A and the small b is on the same chromosome originally does not necessarily mean they are going to be inherited together. Okay, so you know whether you get a big A or a small A in the first gene will have no effect on whether you get a big B or a small B on the next gene. Okay, it doesn't even matter that they are on the same side at the beginning, right? So the person can have a sperm that's like containing big A, small b, small c, or a big A, big B, small c, or big A, small b, big c, right? Big A, big B, big C, small a, small b, small c, right? So on and so forth, right? So how many different combinations are there? Big C, I think I'm still missing one. Help me out here, guys. I think I'm missing this one. No, small a, small b, and big C probably. Yeah. So there are a total of 8 possibilities. Okay. 1, 2, 3, 4, 5, 6, 7, 8. Okay. Not 9, 8, because there are 2 choices here, 2 choices here, and 2 choices here, right? So 2 times 2 times 2 is actually 8. So again, the independent assortment states that how one gene inherits will not affect how the other one inherits. Make sense? Now, in reality, that's actually not completely true. Some traits, if they're really close together, they tend to be transmitted together. But we're just gonna ignore that for now. Anyway, moving on, the law of segregation. Law of segregation. What is that? It says only one of the 2 alleles in a diploid organism will be passed on to each gamete. We know that already, right? Because during metaphase, they separate. So you're not gonna get big A and small a, right? You're just gonna get, either big A or small a, not both. Okay, that's it. Okay, we're gonna move on to number 6. Okay, things are gonna get a little bit intense here now with number 6. So if I'm not making sense, please stop me and ask. I'm just gonna do it on a separate page because not enough space here. Let's do it right here. Okay, so in this question, we're trying to figure out whether it is autosomal dominant or autosomal recessive. Okay, if something is dominant, you only need one allele for the trait to show up. Whereas if something is recessive, you need to have both alleles in order for the trait to show up. Okay, so in a pedigree, if something is shaded, then that means the person has the trait. Whatever trait we're talking about, it doesn't really matter. Okay, so we have to decide based on the pedigree whether the inheritance pattern is dominant or recessive. Okay, so for example, big A, or big A, small a. So that would be the dominant trait. Okay, whereas small a, that would be someone with the recessive trait. Okay, so something is recessive if you need 2 copies in order for it to show up. Okay, for dominant, you only need 1 copy. You can have both, but 1 copy is enough for it to show up. And so let's take a look at the first one. Is the first one dominant or recessive? Okay, autosomal dominant means the trait is carried on an autosome, right? And autosome is chromosome 1 to 22, not including the sex chromosome. Okay, so here is the telltale sign. Okay, you should write this down, put it on a sticky note, right? Along with all the other, cheat sheet that I tell you to, other things that I tell you to put on a cheat sheet. So here, okay, autosomal recessive. Okay, there is something that you can look for. If you see this, it's guaranteed to be autosomal recessive. if these are the only two choices. Okay, so if you see two parents without the tray, having a child with the tray, then it's autosomal recessive. Okay, I'll explain why in a second. So let's take a look at this first one here. Right? We see two parents. They are not shaded in. The dad doesn't have it. The mom doesn't have it. But then the daughter has it, right? The daughter is shaded. So you see two parents without the tray, but then the one of the kid has it, right? Okay, so that's a dead giveaway that it is autosomal recessive. Okay, so this is recessive. Okay. Now let's try to fill it in. Sorry, give me 1 second. So this is autosomal recessive because of what I said. So that means this person is going to be small a, small a. Okay. Now you might wonder, how can two people who don't have the tray end up having someone who does? Well, they could be heterozygous, right? So if the father is big A, small a, and the mother is big A, small a, right? They both carry the small a allele, but they're not showing it, okay? Because it's a recessive trait, right? To show the trait, you need to have both of the recessive allele. So if I do the Punnett square, big A, small a, big A, small a, right? Okay. I assume you saw the video, so you understand this Punnett square thing. You see that there's a 25% chance, small a, right? Now for the brothers, this one's gonna be big A something, but there's no way for us to tell. Okay, they could be big A. There's 25% chance they're big A. There's a 50% chance they are big A, small a. Okay, so that's all we can do for this one. Let's take a look at the next one. Now, if you're not sure, I mean, you can try to look for what I was saying, right? Two parents without the tray, having a child with the tray. So I don't see any of them. Okay, I don't see any kids who have the tray, but the parents don't. You can try to do it like this. You can try to assume. Okay, let's just assume this is recessive. Okay. If I assume this is recessive, then I can say, okay, this person, this guy has it, so it's going to be small a, right? And then this woman also has it, so small a. Now, if the father and the mother are both small a, and we are following this recessive pattern, then that means all of their kids should have the tray, but they don't. So that's how I know this is not recessive. Okay, so I assume this is recessive, but it can't be, but it cannot be. Okay, why? Both parents, right? Well, we can say this, both I1 and I2. Okay, generation 1, number 1 and number 2 has the tray. Have the tray, but none of their kids, none of the children has it. Okay, so it cannot be recessive. So you guys understand? Right? Because if you do the Punnett square, it should be 100% small a for all the kids, but it's not. So that means it must be dominant. It must be dominant. Okay. So big A for sure. I don't know about the other one yet. Big A here and something. Okay. Does anybody want to tell me whether this is big A, homozygous, or big A, small a? Help me out. Let me know you understand what I'm talking about. Is it big A, or is it big A, small a? How do we know? Anybody? It cannot be both. It's either big A, or big A, small a. Okay. Christina said big A. Sita said big A, big A, small a. Okay, let's try to figure it out, right? Okay, let's say they're big A, big A, big A, big A. I'm not saying that's the answer, but let's just say that is, okay? So let's say they're both big A, big A, big A, big A. What does that mean? That means 100% of their kids will be big A, right? Because that's the only thing they could inherit, yes? And that's not true. Because if they are all big A, then that means all the kids would also have the trait, right? But they don't have the trait. So that tells you the parents must be big A, small a, like that, right? So that's the only way to explain this. Okay, you cannot even have one that's big A, and then the other big A, small a. If you do the Punnett square for this, all of the kids will also have the trait. The only way for them to have kids that don't have trait is if they are big A, small a. Okay, so this daughter is going to marry some guy who has the trait, which is big A, small a in this case. And I know it's big A, small a because they also have a kid who does not have the trait, right? Okay. Trait meanings like whatever big A stands for. Okay. So for example, I can say big A is like it's like brown eyes. Okay. Small a would be blue eyes. Okay. It doesn't really matter. It could be other things, right? Okay. So both of them have brown eyes, but then all these people have blue eyes. Right. Big A could be ability to curl your tongue. Small a would be cannot curl the tongue. Right. So there are a lot of different traits, right, that we talked about. All right. Eye color, earlobes, right? Hairline, whether it's straight or widow peak, you know, blood type, all these things. Okay. Anyone have any question about this? I know it's confusing, and it's okay if you have questions. Okay, let's take a brief break, then talk about something else for a second. Next week, next Thursday, we will have no tutorial next week, right? Okay, because you're supposed to work on your lab, and I'll explain the lab towards the end. Okay, now, however, looking at the results of the recent quiz, actually, it's not bad. The recent quiz is, you know, average is quite high, but I'm saying lecture 7 and lecture 8, some of these stuff, it's a little bit confusing. Okay, so we won't have a official tutorial next Thursday, but we can use it for extra help. Okay, so if any of you wants to come to the extra help next Thursday, send me an email. Okay, and then I will do a session. If nobody emails me for extra help, then there will be no class. Okay, so email me by, I don't know, Tuesday or something, and then I will set it up. Okay, I'll make an announcement on eCentennial about the final decision, where there will be extra help or no extra help. Okay. Tutorial for what lecture? Okay, so like next week again, there's no new lecture, right? Okay, no new lecture next week. You just have to work on your lab. But I'm saying next week, since there is no scheduled tutorial, and I feel that people are probably a little bit confused about lecture 8 or lecture 7 with all the codons and stuff like that. So I'm saying if you need extra help, please come. Okay, but when you come to the extra help, you know, be prepared to turn on your mic and actually talk to me, right? Like converse with me, right? So that I can actually help you. Okay. But send me an email though. Okay, send me an email. You can ask me anything in the extra help, anything from this lecture 6, 7, or 8. All right. And you should ask for help if you're confused, because lecture number 9, that is a killer. Okay, that is the hardest lecture of the entire course. Okay, And then number 10 will be very easy. Okay, but we must go over the Himalayas first, and then before we enjoy the very easy lecture 10. All right, so let's just get out of the way 6, 7, and 8, and then we'll see. If no student 5 and below need extra help, if number of student what? Oh, doesn't matter. If one of you need extra help, I'll be there. Okay, but you got to email me. Okay, and tell me what you need to go through. All right. Don't just show up and say I need extra help with no actual questions. Okay, I'm not the one who needs to come up with questions to help you with. You need to tell me what you need help with. Okay. So I'll post an announcement about that. All right. And again, be prepared to turn on your mic. Have some interactions. Anyways, moving on. Next part here. What do you guys think this is? Okay, let's do a poll. We haven't polled for an hour. Let's do some polls. What do you think? If you think is dominant, please choose A. If you think is recessive, please choose B. If you don't know, that's okay as well. Please choose C. Okay. What is going on over here? All right, let's get those numbers up. We only have 8 responses so far. Come on, you can do it. Even if you're not sure, there is an option for you. Call not sure. You can choose that. Anyone else? Last call. Missing 2 people. Do you guys want to lock something in, or are you there? Okay, maybe they're away from keyboard. Okay, so we have more people choose dominant, and then some people choose recessive. Some are not sure. That's okay. Let's read what this purple thing says here. Okay. 2 parents without the tray having a child with the tray. If this happens, then it's going to be recessive. Okay, so look here. First of all, how do you know if someone has the tray or not? If it's shade in, then they have the tray. So this person has the tray. And what about the parents? The parents don't have it, right? So based on this, right, and based on this, we know this must be recessive. Okay. All right, we got to move on. And think more about this. Try to fill in the genotype yourself. All right, we got to move on to the problems. Lots of problems to do. All right, let's go through the monohybrid first. I don't know if you guys had a chance to look through these. Does anybody have a request as to which one you want me to do? Any particular request? No requests? All right. Go ahead.
All of them. Can
all,
you
all of them? All right. I'll do a few. Okay, we won't have time for all of them, but I'll do a few, and then you guys can probably try it on your own, and then come again next week or something for help. Where was I? Okay, right here. The hardest one. I don't know. The hard is subjective, right? Okay. Anyway, we'll just do some. We'll just do the ones I did with the other class. Monohybrid. Monohybrid means we're tracking one tray at a time. Okay. So here we have a black coat color in guinea pig, and that's dominant over white coat. Right? So what we want to do is it's like math. Okay, we want to declare our variable. Okay, so we say big B is black coat. Okay, big B is black, and it's the dominant one, so that's why we use capital letter. And small b is white coat, so we use small letter because it's recessive. Okay, so we write down what the allele represents, and then we want to write out the possible genotypes. Okay, so it's always diploid unless otherwise stated. It's always diploid. Okay, you can count on it. So let's say we have big B, and big B, small b. Right? So these individuals will have black coat. Okay, because one big B is enough to turn the coat black, right? Okay, because that's the dominant allele, and then small b, right? It's going to be white coat. Before we proceed, any question about this? Okay, couple of terminologies in case you forgot. If it's the same allele, we call it pure or homozygous. Okay, if it's not the same, then we call it hybrid or heterozygous. Okay. For the recessive trait is always homozygous. Okay, because that's the only way for it to show up. Right. So if I give you a guinea pig and it's a white guinea pig, you know for certainty that it is going to be small b, small b. If I give you a black guinea pig and I ask you what's the genotype, genotype means the genetic makeup, right? You won't be able to tell whether it's big B, or big B, small b. Okay. So let's take a look at A. We have, what do we have? We have a hybrid black pig and a white pig. Okay, we're gonna breed them. We're gonna make them have babies. So the hybrid black pig, hybrid means different, right? So that's gonna be big B, small b. Right? That's why they can't just say black pig. If they say black pig, you don't know if I'm talking about big B, or big B, small b, right? That's why they have to tell you if it's a hybrid black pig or a pure black pig. So the hybrid black pig is gonna have babies. We put the cross here with a white pig. White pig is small b, small b. And they don't have to tell you whether it's pure or hybrid because white is the recessive color in this case. And so it must be homozygous. Then we can do our Punnett square. And we have the big B, small b from the daddy pig. And then the small b from the mommy pig. And then we're going to cross them. Okay, so there's a 50% chance, 50% chance that the babies are going to be big B, small b, in which case it will be black. And then 50% chance it's going to be small b, which is going to be white. Any questions? Okay. Let's just do one more, just for safety. B, we have a white mated with a pure black. So white by default is small b, cross with pure black, which is big B. How do I know it's big B? Because it's pure. Pure means the same. Okay. We can do the Punnett square. Big B here. So half all the sperm will have the big B, right? Basically, and all the eggs will have small b. Could be the other way around. We don't know which one is the male, which one is the female. It doesn't really matter. So big B, small b for all of them. So 100% big B, small b, black. Okay. That is it. Let's do number 4. Number 4, it looks pretty challenging. The ability to curl your tongue. It's dominant to not being able to curl your tongue. So again, we have to declare a variable. And we should write down what we let the letters represent. So big T means is a roller. You can roll your tongue. And small t is non-roller. All right. Because it says roller is dominant over non-roller, right? So again, you don't really have to do this part if you already understand, but it helps sometimes if you are new to this. Big T, right? Pure roller and hybrid roller, right? Regardless, both of these would be roller. And then small t would be non-roller. Okay, non-roller. So what's going on here? All right, a woman who can roll her tongue. Okay, so woman, circle to represent woman. This person can roll her tongue. So is she big T or big T small t? We don't know yet at this moment. Okay, we just know she's gonna have one big T and then the other one could be another big T, could be a small t. Not sure at this point. But she is going to marry a man. Man is square. And this guy is a non-roller. So non-roller, small t. Okay. And then they're gonna have a kid. The first child has his father, so it's a boy, square, has his father's phenotype. Phenotype means the expression of the gene, right? So if the father is a non-roller and the child is the same as the dad, then he is going to be small t as well. Okay. So one of the small t came from the father. The other small t must have came from the mother. So the mother is going to be big T, small t. Okay, so these are the genotypes of everybody. The answer is right here. B, what is the probability that a second child won't be a roller? So we will do the Punnett square. Okay, so the mother can either have eggs carrying the big T or eggs carrying the small t. Father is just going to have sperm that carries the small t. Okay, we technically don't have to do 2 rows, but whatever, we just do 2 rows here. So we combine them. And you can see there's going to be a 50% chance. 50% chance of having a non-roller. So their second child have half a chance of being a non-roller. That's it for number 4. Are we okay? All right. Let's do another question. Let's do the dihybrid problem. I'm just trying to see which one to do. Dihybrid problem. Okay, let's do this one. Number 4. There we go. Okay, I'll read it to you. In rabbits, black fur is dominant over brown fur. Normal length fur is determined by a dominant gene, and the short fur called Rex is the recessive allele. Okay, so we'll stop there. Let's just focus on this first part here, and you know, we can write down what we have. So here are 2 separate traits, right? We have one trait for the coat color, and we have one trait for the length of the fur. And so black is dominant over brown. So big B is going to be black. Small b is going to be brown. And we're going to have the fur, big N for normal fur, and small n for short fur. Okay? Use capital letter to represent the dominant trait and small letter of the same alphabet to represent the recessive trait. Okay, so diagram a cross between a homozygous black with homozygous normal length fur. Okay, I don't know if that's the male rabbit or not, but it doesn't matter. I'm just gonna pretend it is. And we're gonna cross it with a brown Rex rabbit. Okay, so let's see what's gonna happen. Okay. Homozygous black with homozygous normal length. Can someone give me the genotype for this rabbit, please? Thank you, Moira. That would be big B, because homozygous black and homozygous normal. Cross with the brown Rex rabbit. Brown is small b, and Rex is small n. And you're like, how do you know? They didn't say it's homozygous or heterozygous. Remember, for recessive trait, they don't have to tell you. Okay, if it's recessive, it must be homozygous. Okay, so this is the parent generation. And now they're gonna have baby bunnies. Okay, so what kind of gametes do we have? Let's write down the gametes. Gametes are like sperm and eggs, right? So we have two traits here, and there's only big B to pass on, and there's only big N. So the possible gametes for this one would be big B, big N. That's the only possible gametes. And for the Rex, brown Rex rabbit, it's only going to be big B, small b and small n. Okay. So if I do the Punnett square, okay, I'm going to put big B, big N here, small b, small n here. So 100%, 100% of the F1 generation, right, the first generation, offspring, first generation of offspring, will be big B, small b, big N, small n, which is normal black fur. Anyone have any question about what I've done so far? Please ask now, because it is about to get more confusing. So if you don't understand something here, let's clarify it first. Remember, if you have a question, someone else probably has the same question.
Hi, sir.
Mm-hmm.
Why didn't you do the 2 rows for the square? Like for the
Punnett square?
finding the probability. Yeah.
Let me just clarify the question. If we do 2 rows, are you thinking to put B here and N here like that?
I'm just wondering why you didn't, like, because usually, like, when we were doing the other questions,
Oh,
you
I
did
see
it,
what
and
you
then
mean.
this one you didn't. So I'm just.
Yeah, okay, I understand. We only have to do 1 because there's only 1 possible gametes, right? If there are more possibilities, I would be doing more. Okay, so for example, okay, let's just pause this question for a second, and then let's just say we have a rabbit that's like big B, small b, big N, okay, and then cross with small b, let's cross with big B, small b, big N, small n. Okay, so this is not really related to what we're doing, but I'm just showing you guys on a side, right? Okay, so let's say I have this. What are the possible gametes? Okay, so let's think about the gametes for this guy. Okay, what are the gametes? Well, we can have a big B and a small n, right? Sorry, big B and a big N. And then here is big B, big N again. So same thing. We can have a small b, big N, small b, big N. So for this particular rabbit, the possible gametes would be big B, big N, or small b, big N. Okay. Now, what about this one? What are the possible gametes for this one? Who was it? Was it Saida? I didn't quite catch the name earlier. I think it was
Yeah,
you,
it
right?
was
Yeah.
me. It was Saida, yeah.
Okay. Would you like to try to give me a gamete for this rabbit here?
So would it be big B and then big N?
That's one of them. And then anything else?
And I can also do small b and big N?
Yeah. Yeah.
Yeah.
Anything else?
And then big B and small N,
Yes.
maybe?
Yeah. And then one last one.
I'll be small b and small N.
Exactly. Okay, thank you. So there are two possible gametes for this one, four possible gametes for this one. So when I set up my chart, I am going to have four things here, right? From this rabbit, and then I'm gonna have two things here from this rabbit. Okay, so the number of columns and number of rows depends on how many different gametes we are generating, right? So over here, we only had one possible gamete for each rabbit. That's why we only need like one row and one column, right? And over here, because we have 2 and 4, that's why our chart looks different. Does that answer your question?
Oh, okay. Yeah. Thank you so much. That makes sense.
Okay. Yeah. No problem. Anyone else have other questions before we move on? Okay, so that's done. That's F1. The question also says determine the ratio for, ratio, this is not ration, so ratio for F2 generation. So what does that mean? It means we're gonna take two of the rabbits from the F1 generation and we're gonna cross them. Okay, so we're gonna take a big B, big N, small n, and we're gonna cross it with another one that is from the same generation. Okay, so what is going to happen here? Well, we will also have to write out the gametes. Right? And we just try to do the different combinations, right? We did it earlier. So big B, big N, big B, small n, small b, big N, small b, small n. And it's the same thing here, right? It's kind of like doing polynomials in your math class, right? So the possible gametes are listed here. And it's the same thing for this one. So we're going to have very, very big chart. And we're going to have 16 boxes in total. So 4 by 4. Let's do this. Oops, sorry. All right, so I'm going to put the gametes of one of them here. Okay, for those of you who kind of get this already, I suggest you draw this out as well, because I will show you how to use this to answer questions on the test. Okay, even for those of you who are expert in this, like, yeah, I know how to do this. Just write it down, okay? We'll see how to use this on the test question. Okay, so why don't we fill it in? Okay, I'm going to give you a moment to fill it in, and then I'm going to fill it in too, and then you can compare. All right. And again, for those of you who are very good at this, please double check my work. Make sure I'm doing it right. There we go. Okay. Now, does anyone want me to explain like how I got some of these things? Like, is there a particular one that you're not sure how I arrive at that answer and you want me to explain? Please let me know. All right. So help me out here. Someone tell me what would the sorry, does it need to be exact order? No, but we typically put the capital letter first. Okay, like you could have put like NNBB, that's fine. It doesn't really matter, I guess. Is that what you're asking, Sita? Or am I misunderstanding your question? Okay. Someone tell me what this is. What is this this rabbit gonna look like? Oh, sorry, this is not a highlighter. What is the color and the fur length of this one? Big B, 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 N, N. Big B, big B, big N, big N. What's the trait there? Anybody know? Is it black? Is it brown? Is it normal? Is it short? What is it?
Black and normal length fur?
Yep, that is correct. That is normal black. Normal black. Okay. So I want all of you, and hopefully you guys are really writing this down, to find everything else in this table that's also a normal black. Okay, so for example here, right? This is also normal black. Black because you have big B, and then you just need a one big N for it to be normal. So highlight everything that's normal black and tell me what you get in total. How many normal blacks are we gonna get in this box? Only two, really? This one's almost not all. This one is also normal black, right? Maybe more than 4. Yeah. This one's also normal black. Normal black. Basically anything that has a big B and a big N, that's normal black, right? So all the ones that I have colored here, that's going to be normal black. And if I count it 1, 2, 3, 4, 5, 6, 7, 8, 9, that is 9 out of 16 of the boxes are going to be normal black. Does that make sense? Have I convinced you that it is 9? If not, let me know. I can be more convincing. Okay, let's find another genotype. Okay, let's take a look at this one. This one is big B, small n, right? So big B means it's black. Small n means it's short. So this is short black. Okay, let's find all the short black. Do we have an other short black? If so, how many others? How many in total, I should say? Yeah, Sierra says 3, that is correct. There is the short black. There's another short black right here. Okay, so 3 out of 16 is short black. Okay, we can color another one. This one would be normal brown, and normal brown again. So 3 out of 16 is normal brown. And very, very rarely, in this case, you will have short brown. Okay, short brown. 1 out of 16. Short brown. Okay. So that's the classic 9:3 to 3:1 ratio. If you start with purebred parents and you carry on to the second generation. Purebred means the parents are homozygous everywhere, right? So you can see the parents, they're homozygous for both traits. And then if you carry it all the way to second generation, you will always get 9:3:3:1 as your ratio. Now I'm going to show you what you will see on a test, on the quiz. Okay, where's my, right here, okay. We actually have a lot of questions, but we don't have time. We have to keep going. Maybe we'll do some of these things later. Where's the question? Okay, here is a potential question. All right. I'll read it to you. True breeding. True breeding means purebred, right? Purebred. Stalks of peat plants are crossed. One parent has red axial flower, the other has white terminal flower. All F1 individuals have red and axial flower. Okay, so here is the thing. When you cross, you should write this down. When you cross two parents that are purebred, the trait that ends up showing up in the F1 generations, those are the dominant trait. Okay, let me write that for you. When two individual, in this case, the individuals are plants, when two purebred individuals are crossed, purebred, the traits that show up in F1 are the dominant trait. Okay. So just by reading it, like red and white, I have no idea which one is dominant. Right? Is it axial or terminal? Terminal means the flower is at the end. Axial means it's like coming off from the branch, right? Okay. So then, you know, how do you know which one is dominant and which one is not? And in this case, we have to look at the F1 generation. So all F1 will have red and axial. That means big R is dominant. And small r is going to be white. Okay, axial. So big A is axial. And small a is terminal. Okay, I'll come back to this slide in a second. I just want to show you that over here, where is it? Over here. You see, we have the black normal crossing a brown Rex, and then in the F1 generation, all of them will be normal black because normal black is the dominant trait, right? Okay, normal and black. So if you cross two purebred parents, whatever shows up in the F1 generation, that's going to be the dominant trait. Okay. All right, so back to this question. The genes for the flowers, colors, and location are sorted independently. If 1,000 F2 offspring results from the cross, how many of them would you expect to have red terminal flowers? Okay, so on a test, you might try to do this by writing big R, small r, big A, small a, and then you will basically take a lot of time to do the question, right? Yeah, but remember, this is only 1 mark. Right? So you don't want to be spending like 5 minutes trying to create that chart, right? So what you want to do is, well, it's good if you memorize the 9:3:1 ratio, but if not, what would be a good idea is to have this on your help sheet as well. Okay, your cheat sheet, right? And it's okay to have a cheat sheet. If this was a face-to-face test, this question would not be a multiple choice question. This would be a actual handwritten question worth like 4 marks or something. But what you can do is you can just paste it here, right? You can refer to the chart, right? So we are looking for, what are we looking for? We are looking for how many plants will have red and terminal flowers. Okay, so red means I have to have at least one big R, and then terminal flowers. Terminal is a recessive one, right? So I have to have like small a, small a. Okay, so big R something and small a, small a. And if you look at it, only 3 of them are like that, right? Okay, it's just different letters, but it's the same concept. So that means 3 out of 16 times 1,000, right? I don't know what that is. We got to plug that into the calculator. That's going to be 187.5, right? So the closest answer is E. Okay. Now it's not going to be exactly 187, right? It's just like this, right? You flip a coin 10 times, you expect to have 5 heads, 5 tails, but chances are you're not going to get exactly 5, right? Because what happens in real life versus statistical, like theoretical statistics, it's different, right? So like statistically, you're supposed to have 180 and a half of a flower being like this, but when you actually do it, right, it's just going to be a number that's close to it. Okay. So does everybody understand how I get 190 and how to get it using the chart without having to make the chart? Any follow-up questions?
Sir, will you be posting this, like the little chart that we did for the other question on
Yeah,
the-
sure. I'll post it. Yeah.
Okay, thank you.
Okay. Still have a couple other things to do, but let's do another one here. In a cross of this and this, what is the probability of producing a genotype of this? Okay. So again, a little bit of statistics background in case you haven't done it in math yet. So let's say I have a coin, right? I have a coin, okay, and I have a dice, okay. So what's the probability? Probability of getting head plus a 4. Okay. So head and a 4, right? So to do this, we have to find out the probability of getting a head. Probability of getting a head when you flip the coin is half. Okay. Head and 4. What's the probability of getting a 4 on the dice? Anyone? Roll a dice. What's the chance of getting the number 4? 1/6. That's correct. Okay. There's only 1 4 on the dice, right? Out of 6 possibility. So what's the probability of getting a head and then a 4? You would multiply the individual probability together. So it's 1 out of 12. Okay. My point is this. If you have a bunch of independent events and you want to find out the probability of all of them happening together, you multiply their probabilities together. Okay. How does it relate to this question? All right, so we have 3 genes, right? Okay, we have A, B, and C. And according to the law of independent assortment, whether you get a big A or a small A will not affect whether you get a big B or a small b, so on and so forth. Okay, so we must evaluate each gene individually. So let's take a look at AA here. So if I get big A, small a, and I get big A, small a again, right? Because that's what happens for both the parents. And I do the Punnett square, I will see that there is a quarter of a chance that I will get big A, right? 1/4 of a chance for the big A, Okay, we can do the same thing for B. It's the same Punnett square, but instead of having A, we'll do B's. And you will see that it's also a quarter. Same thing for C, so also a quarter. So I have to multiply all of them together, and that's 1/64. Okay. I mean, I can write it out for clarity. Big B, small b, big B, small b. Okay, so and other quarter for this. And then same thing for C. Okay. So quarter, quarter, and quarter multiplied together, 1/64. That's the answer. Is that okay? All right, let's try this one. Okay. These are all real questions that you can see on the test, right? On the quiz. Please try this. Go ahead. I have one answer out of 15. That's not good. Okay, I'll do it. That's okay. All right, so here we go. How many unique gametes, right? Okay. So there are okay, before we do the number of gametes, how many genes are we talking about here? Give me a number, guys. How many genes? How many genes here? Yeah, 5 genes, right? Okay, A, B, C, D, E, 5 genes. All right, 5 genes. So let's take a look. Do you have a question, Sierra, or just accidentally unmute? Okay. 5 genes here. Okay, so let's take a look at the AA. How many possibilities are there? Okay, you can either inherit the big A or the small a, right? So there are 2 choices for gene A. How many choices for gene B? Well, I hope you can see it is also 2 choices. You can get big B or small b. How many choices for gene C? Yeah, just 1. You either get big C or big C. There is no other choices, right? For gene D, 2 choices, big D or small d. Gene E, 1 choices. You multiply all of them together, it is 2 times 2 times 1 times 2 times 1, which is 8. Okay, which is 8. Alex, you mentioned 32 earlier. Okay, it would be 32 if it's like this. Okay, if they're all heterozygous, then 2 choices, 2 choices, 2, 2, 2. So 2 times 2 times 2 times 2 times 2, that's going to give you 32. Is that okay? Will you guys be able to handle this on the quiz? I sure hope so, for your sake. All right. What else should we do? Okay, let's do one more here. The pedigree below shows some of whose members exhibit the dominant trait woolly hair. Okay, so black circle or square are people with woolly hair. I don't know what that means, but it doesn't matter. Okay, those are the people with the woolly hair. And we want to know, what is the likelihood, what's the probability that the children of generation 4, number 3 and number 4, so these 2 people, what's the probability of their kids also having woolly hair? Okay, so let's find out. So big W is woolly, and then small w, we just call it normal. So individual 4 here in generation 4, that's what this means. No woolly hair for this lady. So she will be little w, little w. The husband, who is shaded in, is going to have woolly hair. But the question is he homozygous, as in big W, or is he heterozygous? He might be big W, small w. How do we know? Well, we're going to find out from his parents. Okay, so these are his parents. And the mother does not have the woolly hair. So she is small w, small w. Okay, so for that reason, we know this guy must be heterozygous because the little w came from the mother, right? Now that we have the genotype of these two people, we can go ahead and do the Punnett square. Okay. And we'll see there's a 50% chance of big W, small w, woolly hair. Okay. The answer is 50%. Okay. That is that. Let's move on. Let's do I'll skip the incomplete dominance and codominance for now. We will do the sex link. Okay, this is explained in the lecture, so you can take a look, and if you don't understand, you can ask me about it later. But sex link is usually a little bit more confusing. So let's do this one. Something is sex linked if it is transmitted on the sex chromosome. Okay, you have 23 autosomes, so those are autosomal dominant or autosomal recessive. If it's passed on the X chromosome, then it's X-linked, and if it's passed on the Y chromosome, then it's Y-linked. If something is Y-linked, only males can get it, right? So fruit flies, the eye color is sex-linked, right? And if they have XW allele, that's the allele that will contribute to white eye. All right, so let's write down all the possibility here. Let's look at the female or the male first. Let's do the male first. Male fruit flies. Okay, so it could be a XY. So that would be a red eye male. Okay, red eye male. And the male could be a XWY. Right? So this is the YI gene. It's an X-linked gene. It's carried on the X chromosome. That's why we have a superscript here on the X. This would be a YI male. Okay. It's crazy. The people who work with flies, they can make them any eye color, rainbow eye color, red, orange, yellow, green. If you just go on Google later and search like fly eye colors, rainbow, then you can see all the flies lined up, right? Starting with the red, which is the most common one we see, and then you can mutate them to give them like any colors. Things we do to flies. Female. We can have XX. Is this red or white? Both of them are normal, so it's red. We can have a XXW. Is this red or white? Anybody? Any takers? Yeah, that's going to be red eye as well, because there is one normal copy, right? So the normal copy will override the recessive one. But this would be a carrier. We call it carrier. And then we can have a XWXW. That's going to be a white eye. Okay. So let's take a look at the first question. We have a white eye female. So white eye female. It's going to cross with a red eye male. Okay, white eye female, red eye male. What's gonna happen to the baby flies? Let's find out. Gonna do the cross here. XXW, XXW, XWY, like that. Okay. So, you know, you will have a 50% chance of getting red eye female. 50% chance of getting white eye male. So in other words, all the females will be red eye. All the males will be white eye. All right. Any questions? Okay. We are running out of time. Again, you just email me if you want to come next week, and then we'll take up the rest or something. Okay, you just have to let me know. I want to spend the last little bit of time talking about the lab. Okay, give me a second. I will post the link after class, but let's just take a look here. Under content, you can go to virtual labs, and then we're doing lab 2. Right? It's 10%, easy way to boost your marks. Okay, you want to watch how to use and care for a microscope? There is a link to a virtual microscope. Okay, and then this is the actual lab itself. Okay, so here's the virtual microscope. I don't think it works too well on the phone, so make sure you do it on a computer if possible. So a lot of people just skip all these things and then go right to the questions. That's not a good thing to do. Okay, read it because it tells you what to do. Okay, so because there's a lot of instructions here and there, I don't want you to miss a question. So all the questions are highlighted in yellow for you. Okay, so first part is just labeling the microscope. That should be easy. Here it will tell you exactly where to click. Okay, so this is the simulation for the microscope. Again, like I know it's not like the real thing, but it's the best we could do. Okay, so I'm gonna click on explore. I'll move this thing out of the way first. Explore. And it will tell you, right? Click on explore, click on the slide box, you just follow it. Okay. So first thing you're gonna have to do is you're gonna have to look at the letter E. Okay, it's out of focus right now, so you have to play with these things. And the description will tell you which one to move around to bring it into focus. Okay. So the question here says, center the image and take a screenshot of the letter F 4 times, 10 times, and 40 times. Okay, so 4 times, 10 times, and 40 times. I want you to take a picture. You can use your phone. You can use the snipping tool, right? Okay. When you take snipping tool, though, don't take the whole screen like this. Okay, and then paste it into the document. Okay, just do the microscope part. Okay, just like this, and then you can copy that into the document and resize it. Okay, don't give me your whole screen, and definitely do not give me a link to go to like your Google Drive or something. Okay, just paste the document into the just paste your picture directly into the document. No links. Okay, it's much easier for me to mark that way. So that's pretty straightforward. A lot of the questions involve taking pictures. Okay, and then this one, I'm going to give you the answer to this one because every year many people get it wrong, and it's just easier to mark if you get it right. Okay, so here it says, what is the relationship between the image, which is what you're seeing, and the object? Okay, so let's go back here. This letter E. Okay, let's open up the floors right now. how would you describe the relationship? What's the relationship between this E and the actual normal E that you put under the microscope? What would you say? It's okay if you get it wrong. Okay, I'll correct you and I'll tell you what the right answer is. But I just want to know what you guys will say. Yeah, okay, upside down, right? Many people will put upside down as the answer, but that's not the full answer because this is not just upside down, right? It is upside down and let me show you what's upside down. Right? This is E. Upside down is this. Okay, that's upside down. But is this upside down?
It's the mirror image, like inverted mirror image, I think.
Okay, yeah, a lot of people also say mirror image, but mirror image would be like this. Like if I put a mirror right here, mirror image would be like this. That's mirror image, right? Okay, that's not really mirror image, right? Okay. So it's a combination of, thank you. Okay, it's 0 is correct. It's upside down and reflected. Okay, rather than saying flip, we can say reflected. Okay, so it's upside down and backwards, right? So the correct word for that is inverted. Let me type that into the chat for you. It is upside down and backwards. Okay. That means inverted. That's the relationship. So if I take the slide and I shift it to the bottom left, then your image is actually going to be moving to the top right. Okay. If I move it to the bottom left, image goes to the top right. It's always going to be opposite and backwards. Okay. So down becomes up, left becomes right. You all understand what to put here? Okay, now remember, these are like 3 marks question. So don't just answer one part and not the other. The other question is, besides the letter E, give 2 other letters that would also be appropriate for this activity. Explain your reasoning. Okay? What other letters will be a good choice to demonstrate this concept of, invertedness? Yeah, G is a good one. Yeah. How about S? Can I do S? Is S a good one? Yeah, S is not good, right? Because if you rotate S, it looks exactly the same, right? So you got to think about what letter to use. All right. That's good. Okay, these are very simple stuff. Again, you got to read all these things. Okay, like people lose marks because they just don't read stuff. They just skip right to the question and then hope for the best, right? You have to read it because the answer, the description is all here. Okay. And there is going to be a sample calculations here. I don't know why these like upside down question marks is showing up. Okay, like just ignore them. They're not supposed to be there. Okay, just ignore them. And then there's a sample calculation. Okay, so I'm going to click on that. I'm going to mute myself so I don't hear myself talk. So this is a sample question that shows you, if you have a 40 times magnification with this field of view, what would be the field of view at 100 magnification, right? And then I will go through all the math that shows you how to do it. Okay, so watch this and then you will know how to do it. But here's the problem. Here's the problem. The people in the past, and you'd be surprised how many people did this. They just wrote down exactly what I did in the video and put it here. And that would be wrong because those numbers are not the same for this question. Those are just numbers that shows you a sample. When you do this question, it's going to be different numbers. Okay, so you must make sure you use the same numbers. Okay, why would I make a video and ask you to copy exactly there and put it here for 3 marks? I wouldn't do that, right? Okay, so make sure you use the right numbers. All right, so that's one thing to watch out for. This question is asking you, look at this picture, right? And here is one onion cell, and I want to know how long it is. Okay, so the answer is not going to be exact, but there is a range of answer that I'm going to accept. Okay, as long as you show me the math and you can justify your reasons, and it's logically sound, you will get the answer. Okay, so don't freak out if you got one number and your friend got something a little bit different, because what you get here is going to be based on what you get at number 6. And the number you get at number 6 is going to be based on how you interpret this picture. Okay, again, there are no definitive answer. There's a range of answers that are going to be acceptable. Moving on. These are very straightforward stuff. You have to find a picture of a cell in anaphase. So make sure you find that individual cell and just paste a single cell here, not a whole field of cells, and ask me to find it. Okay, you find the one that's in anaphase and you paste it here. And I think everything else is pretty self-explanatory. Again, 10%, excellent way to boost your grades. Okay, get it done this weekend and forget about it. Okay, don't wait until last minute. That's usually how people, hand in suboptimal quality work because they procrastinate. So try to get it done. Okay, that is it for this week. Thank you for coming. And like I said, the tutorial next week, it's for extra help, but you must send me an email along with the topics that you want to cover. Okay, otherwise I'll see you in 2 weeks. Have a good weekend. Bye for now. Oh, I already got an email requesting for extra help. Excellent. Okay, keep them coming. Bye now.